Oh, welcome back. This is the second quiz in glycolysis, and I hope you enjoyed this. We're going to go over some questions and we're going to solve them. And I really recommend you do it yourself. Pause the video, try to guess the answer, and see if you got it right. It will be a good way to practice and learn. Alright, let's get into it. Question 1. Under anaerobic conditions, pyruvate is converted to lactate through the process of fermentation. Why is this conversion necessary, and what would happen if it didn't occur? So it has two parts. Firstly, why is this conversion necessary? So first answer is it regenerates NAD plus for glycolysis to continue in the absence of oxygen. Glycolysis would cease due to NAD plus depletion. That makes sense. It generates ADP through substrate level phosphorylation. That's not true. It does not. It prevents the accumulation of toxic intermediates. That wouldn't make sense because conversion to lactate is toxic itself because lactate is acidic and it's toxic. And the fourth one is supplies reducing equivalents for oxidative phosphorylation, and the electron transport chain would be impaired, leading to decreased ATP production. That is not true because although it does supply NADH, right, it wouldn't be impaired. And therefore the answer should be 1. It does regenerate NAD plus so that glycolysis can continue. And the answer should be A. Question 2. During intense exercise, skeletal muscle relies on glycolysis for ATP production. What advantage does glycolysis provide in this scenario compared to oxidative phosphorylation? Number 1, it produces ATP more rapidly. That is true. Glycolysis can sustain ATP production during high intensity activities. And that is also true because during high intense activities in the skeletal muscle, you run out of oxygen, right? But you still need ADP. And you use some other way to get it. And you basically have to use glycolysis more than you do oxidative phosphorylation because you don't have oxygen to use. It does not produce ATP in larger quantities, therefore second one's wrong. It generates ADP with higher energy content. That does not make sense. And fourth, both glycolysis and oxidative phosphorylation, they both produce ATP without generating lactate. Therefore, it cannot be an advantage when glycolysis itself does not produce lactate, right? And also the second part, glycolysis minimizes the production of metabolic byproducts. It does not minimize the production of metabolic byproducts. Actually, you could argue that it increases lactate production during intense exercise because of anaerobic respiration. Therefore, the answer is 1. Question 3. What would happen if aldolase had reduced enzymatic activity? Alright, let's think. Firstly, what does aldolase do? Well, aldolase catalyzes the reaction of cleaving fructose 1,6-bisphosphate into DHAP and GA3P, right? It was the fourth step in the glycolysis. So if we had reduced enzymatic activity, what would happen? Well, let's see. First one, glycolysis would be unaffected. And no, obviously it would be affected because you have reduced activity. So it's not the first one. Second, conversion of glucose 6-phosphate to fructose 6-phosphate would be impaired. This enzyme does not catalyze that reaction, so this does not make sense. Why it would do that directly? So it's not 2. 3, the production of ATP during glycolysis would be significantly reduced. That could make sense, yeah. And the fourth one is the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate would be inhibited. That does not make sense because this, again, does not catalyze that. It would not inhibit it directly. So the answer should be number 3. Question 4. Glycolysis is regulated by various allosteric effectors. If a cell is low in energy and needs to increase ATP production, which allosteric effectors would be expected to activate key glycolytic enzymes? So in this case, we want to activate glycolysis. You want to increase the ATP production. Therefore, what would activate the glycolytic enzymes? So let's remember from our lessons. ATP, remember, it actually inhibits certain enzymes. And that includes PFK1. So it's definitely not ATP because it would inhibit. It's also not citrate because that would inhibit as well. So it's AMP. And number 4 says 2 and 3. It's wrong because although AMP is correct, citrate is wrong. So the answer is 2 AMP. Question 5. A research study focuses on the role of a specific microRNA in the regulation of glycolysis. Knockout mice lacking this microRNA exhibit increased glycolytic flux and enhanced tumor growth. What conclusion can be drawn from these findings? So we have a microRNA that is involved in the regulation of glycolysis, and knockout mice, which means mice that don't have this microRNA, right? The gene has been knocked out of them. They exhibit increased glycolytic flux, right? What this means is that without that microRNA, the mice have increased tumor growth and glycolysis function. Therefore, the microRNA must have had the opposite effect, right? Because removing this increases tumor growth. Therefore, this prevents tumor growth. So it has to be number 1, which is microRNA inhibits glycolysis and suppresses tumor growth. It cannot be the second one because without it, you have these things, right? And the third one obviously has an effect on glycolysis or tumor growth. It already says that in the question. And the microRNA regulates an alternative metabolic pathway instead of glycolysis. We didn't talk about that, so you cannot conclude that from these findings. That's the conclusion of the first 5 questions. Thank you for watching. Watch part 2 for the remaining 5 questions.